picture this. You are sitting down at your childhood dinner table. You're staring down at a plate of pale, mushy, just aggressively boiled broccoli.
Oh, yeah, we've all been there.
Right. And a parent is standing over you, armed with that classic parental logic, insisting, eat it, it's good for you. And you probably stubbornly push it around your plate, trying to hide it under your mashed potatoes.
Or hoping the dog might subtly make it disappear.
Exactly. Well, I have some totally fascinating news for you today. Your parents were actually entirely right about that broccoli being an absolute powerhouse of health. But by boiling it to death on the stove, they were actually destroying its greatest superpower.
Yeah, completely destroying it.
So, welcome to the Deep Dive Lab. Today we are exploring a biological mystery that sounds like, it sounds like a plot device from a science fiction film, but it is happening right inside your refrigerator crisper drawer.
It really is. It is one of the most elegant defense mechanisms in the natural world. We are looking at a specific chemical locked inside cruciferous vegetables that has the proven ability to act almost like a targeted pharmaceutical in the human body.
Which is wild to think about, just sitting in your fridge.
Right. And we are seeing clinical evidence that it can fundamentally alter everything from your fasting blood sugar levels to the actual physical structure of your brain chemistry.
And to guide you through this, we have pulled together just a fascinating stack of research. We are looking at a landmark 2025 study from the journal Nature Microbiology, along with some incredibly compelling clinical trials on psychiatric health from the Journal of Clinical Psychiatry and, well, decades of epidemiological data.
It's a lot of ground to cover.
It is. So our mission today for this deep dive is to figure out how a simple vegetable compound pulls off these massive physiological changes, and why unlocking this chemical requires a very specific sequence of events.
Yeah, it is a literal chemical time bomb.
A time bomb. And it is entirely triggered by destruction. Okay, so let's unpack this. Because to understand how this superpower actually works, we first have to look at the massive headache it caused researchers for decades.
Right, so the scientific community knew they were onto something with cruciferous vegetables. Broccoli, Brussels sprouts, cabbage, cauliflower. The epidemiological data was just impossible to ignore.
Because people eating them were so much healthier, right?
Exactly. When you look at large populations over long periods, the folks who eat a lot of these vegetables consistently show dramatically lower rates of various cancers and significantly better cardiovascular health, too.
So the consensus was clear. Eat these vegetables.
Yeah, eat them. But isolating the exact biological reason for why they were so protective was a completely different story. It was super frustrating.
Because when researchers actually opened up the intact plant, right, and peered inside its cells to find this miracle compound, it just wasn't there.
Nope, nowhere to be found.
Instead, they found a different compound called glucoraphanin. And the really frustrating part about glucoraphanin is that it is biologically inert.
Yeah, totally inactive.
If you isolate it and put it into a human body, it basically does nothing. It just passes right through you.
Which brings us to the plant's brilliant evolutionary trick. It is essentially a microscopic chemical heist.
I love that phrase, a chemical heist.
It really is. So the plant produces this inert glucoraphanin, but it stores it in a very specific cellular compartment. And completely separated from that, in a totally different compartment, the plant stores an enzyme called myrosinase.
Okay, so they are kept physically apart.
Exactly. As long as the plant is whole and undamaged, just sitting peacefully in a field or resting on a grocery store shelf, these two compounds never meet. They're kept in strict physical isolation.
I absolutely love this mechanism. Well, think about how a glow stick works.
Oh, that's a great way to put it.
Right. You hold a plastic glow stick in your hand at a concert, and you have two separate chambers of liquid inside the tube. As long as a barrier holds, nothing happens. It's totally inert.
Yeah, just a plastic tube.
But the moment you bend and snap the stick, you break that internal glass barrier, you cause physical damage, the chemicals flood together, they mix, and suddenly, boom, you have this highly reactive glowing light.
That is exactly it. The plant is executing that exact same strategy, just on a molecular level.
So the snap of the glow stick is the vegetable getting damaged.
Precisely. The snap happens when the plant's cellular tissue is physically damaged. In nature, that damage occurs when, say, a hungry caterpillar takes a bite out of a leaf.
Oh, wow.
Yeah, so the cellular walls rupture, the myrosinase enzyme spills out, and it mixes with the glucoraphanin. The enzyme immediately acts as a catalyst, and it literally tears a specific sugar molecule right off the glucoraphanin.
And that violent chemical reaction creates a highly reactive, incredibly potent compound called sulforaphane.
Sulforaphane, that is the magic word for today.
It's basically a chemical weapon meant to deter bugs, right?
Yeah.
But in human beings, it acts as a cellular superhero.
It really does.
But this brings us right back to that mushy broccoli on your childhood dinner plate and the great cooking conundrum. Why was that boiled broccoli chemically dead?
Well, it comes down to the extreme fragility of the myrosinase enzyme. Proteins and enzymes change their physical shape when they are exposed to heat.
Like boiling water.
Exactly. Think of how a raw egg turns solid when it hits a hot frying pan. The research shows that heating broccoli to just 70 degrees Celsius, which is about 158 degrees Fahrenheit, for a mere 10 minutes completely denatures the myrosinase.
So it melts it.
Yeah, it melts the shape of the key so it no longer fits into the lock. You lose about 95% of the active enzyme.
So if you are vigorously boiling or aggressively steaming your broccoli until it's soft, you're snapping the glow stick, but you've already melted one of the chemicals needed to make it glow.
That's spot on. You end up swallowing the inert precursor, glucoraphanin, and you miss out on the sulforaphane entirely.
But isn't there something about our gut bacteria that can help with this?
Yes, there is. You do retain a tiny bit of hope in the form of your gut microbiome. Some specific human gut bacteria actually produce their own version of the myrosinase enzyme.
Oh, that's handy.
It is, and they can do a fraction of the conversion work for you. But human gut bacteria can only convert a very small percentage of what the plant could have done itself if the enzyme had survived the cooking process.
Which raises a massive logistical question for the scientific community. If this compound is so incredibly fragile and, so easily destroyed by a common kitchen mistake like oversteaming, how did scientists finally manage to prove what sulforaphane can actually do inside the human body?
They had to get very precise with the delivery mechanism. They began using highly controlled, standardized extracts where the sulforaphane was either pre-converted or stabilized.
So taking the cooking variable totally out of the equation.
Exactly. And that precision opened the door to some truly groundbreaking clinical trials. One of the most significant recent breakthroughs is a fascinating study focused entirely on prediabetes.
Okay, this is the 2025 study from the journal Nature Microbiology, right?
Yes. Led by Dr. Anders Rosengren and his team at the University of Gothenburg in Sweden.
Got it.
They designed a beautifully rigorous 12-week double-blind placebo-controlled trial. They brought in 89 participants who were all overweight or obese, and importantly, they all had elevated fasting blood sugar levels.
So these are folks sitting right on the metabolic cliff edge of full-blown type 2 diabetes.
Exactly. Half of them received a daily standardized dose of sulforaphane, and the other half received a placebo. And neither the researchers nor the patients knew who was taking what.
So here's where it gets really interesting. Because the results at the end of those 12 weeks weren't just a minor statistical blip.
Not at all.
The participants actively taking the sulforaphane experienced a significantly greater reduction in their fasting blood sugar levels compared to the placebo group. It was actively pulling them back from that metabolic cliff edge.
And the researchers drilled down even further into the data to see who benefited the most. The difference was remarkably profound in a specific subgroup.
Who was that?
Individuals with mild age-related diabetes, a slightly lower body mass index, and a naturally lower baseline of insulin secretion.
Wow, that's specific.
It is. But Dr. Rosengren's team took it a step further. They collaborated with a microbiome expert, Professor Frederick Beckhead, to analyze the gut flora of the participants throughout the trial.
Because even with a standardized supplement, those resident bacteria in your stomach are still playing a role.
They are acting as active participants. The researchers discovered that specific strains of gut bacteria interacted directly with the sulforaphane treatment.
Wait, really? How does that work?
Well, when certain bacteria were present in a patient's gut, they actually metabolized the compound in a way that enhanced the blood sugar improvements.
That is incredible.
It is a brilliant example of a direct three-way line of communication between a plant compound, your microscopic gut flora, and your overall metabolic health.
But I have to push back here because, as someone reading these studies, it's easy to get lost in the weeds.
Sure.
How can one microscopic vegetable compound do all this heavy lifting. Is sulforaphane just acting like a chemical sponge, floating through the bloodstream and soaking up excess blood sugar and toxins until it's full?
It is a very common misconception, but no, it is absolutely not a sponge. To understand sulforaphane, we have to look at the difference between a direct antioxidant and an indirect antioxidant.
Right, lay it on me.
A direct antioxidant like vitamin C works on a 1-to-1 ratio. It finds a damaging free radical, neutralizes it, and in the process, the vitamin C molecule is destroyed.
So it gets used up.
Exactly. It is a sponge that gets saturated very quickly. Sulforaphane operates entirely differently. It is an indirect antioxidant.
Meaning what exactly?
It doesn't neutralize toxins itself. Instead, it enters your cells and activates a highly specific genetic pathway known as the NRF2 pathway.
Okay, let me translate this because I think the sponge analogy needs to be thrown out the window entirely.
Please do.
Sulforaphane isn't a sponge, it's a biological fire alarm.
Ooh, I like that.
Right. Imagine your cell is a building and oxidative stress is a fire breaking out. A direct antioxidant, like vitamin C, is a bucket of water. You throw it on the fire, you put out a small patch of flames, but then the bucket is empty. The water is gone.
Yep, the bucket is useless after that.
But sulforaphane doesn't fight the fire itself. It walks right into the cell, walks past the flames, heads straight for the NRF2 protein, and pulls the fire alarm.
Now, the fire alarm analogy captures the mechanism perfectly, because when sulforaphane pulls the alarm by activating NRF2, the cell responds by turning on its entire internal heavy-duty sprinkler system.
The sprinkler system being our genes.
Yes. NRF2 is a transcription factor. Its entire job is to travel deep into the nucleus of the cell, bind to the DNA, and tell your genes to start mass-producing a massive cascade of phase 2 detoxification and antioxidant enzymes.
And these aren't just tiny buckets of water. What kind of enzymes are we talking about?
We are talking about fundamental cellular protectors like heme oxygenase 1 or HO1 and NQO1.
Sounds heavy duty.
Very. These enzymes are cellular factories. They don't just neutralize one free radical, they systematically dismantle thousands of them.
Wow.
And unlike that bucket of water that gets used up instantly, these enzymes remain active in your cells for days, continually sweeping up oxidative stress and repairing cellular damage.
This is a long-lasting effect.
Exactly. Sulforaphane doesn't just put a Band-Aid on the cell, it initiates a fundamental reprogramming of the cell's own defense infrastructure.
And because it fundamentally changes how cells defend themselves on a genetic level, the discoveries around sulforaphane haven't stopped at blood sugar and prediabetes. The research is moving into some of the most complex, hard-to-reach areas of the human body, specifically the brain.
Yeah, the neurological applications are truly pushing the frontier of nutritional psychiatry. The brain is an absolute energy hog. I mean, it consumes about 20% of your body's oxygen.
That is a massive amount.
It is, and because it burns so much energy, it produces a massive amount of metabolic exhaust, which we call oxidative stress. If that exhaust isn't cleared out, it damages brain cells and disrupts neurotransmitters.
Which leads to cognitive issues.
Right. To see how sulforaphane could help, researchers conducted a fascinating 24-week double-blind, placebo-controlled study in Hunan, China. It was published in the Journal of Clinical Psychiatry.
Okay, what were they looking at?
They were looking at patients diagnosed with schizophrenia.
Wait, really? This feels like a massive leap from managing blood sugar. How did they set up a trial for a complex psychiatric condition using a broccoli compound?
Well, they gave these patients high daily doses of a standardized sulforaphane supplement, specifically 1,700 milligrams of an extract called Avmacol Extra Strength.
Okay, and were they taking this instead of their normal meds?
No. They administered this right alongside the patient's standard prescribed antipsychotic medications. Now, schizophrenia often involves what psychiatrists call negative symptoms.
Meaning things that are absent,
Yeah.
right? Not the hallucinations or delusions.
Exactly. Negative symptoms are things like severe apathy, a lack of emotional expression, social withdrawal, or just a total loss of motivation.
And those are hard to treat.
Incredibly hard. These specific symptoms are notoriously stubborn. Standard antipsychotic drugs, which usually target dopamine receptors, often really struggle to alleviate them.
But when they added the sulforaphane into the mix, basically pulling that cellular fire alarm in the brain,
The results were striking. The group taking the sulforaphane extract alongside their medication showed a significantly greater decrease in those stubborn negative symptoms compared to the group taking the placebo.
that is mind-blowing.
And what is crucial to understand here is the timeline. The most prominent difference between the two groups didn't happen overnight. It emerged right at the 24-week mark.
Which makes perfect sense when you think about the mechanism. It's not a stimulant. It takes time for those phase 2 enzymes to clear out the oxidative exhaust and rebuild a healthier cellular environment in the brain.
Exactly. If we connect this to the bigger picture, we are looking at a monumental shift in how science views food. We are aggressively moving away from the generic mid-century dietary advice of simply, eat your veggies because they have some vitamins.
Right, the classic parental line.
Yeah. We are entering the era of precision nutrition. Dr. Rosengren heavily emphasized this in his prediabetes study. I mean, prediabetes affects up to 10% of the Swedish population and tens of millions more globally.
It's a massive issue.
It is. Sulforaphane is no longer viewed as just a general health booster. It is being seriously evaluated as a targeted, functional medical intervention to actively disrupt a disease pathway before it destroys a patient's metabolism.
And that precision goes all the way down to the deepest level of our biology, our DNA. We really have to talk about epigenetics and cancer, because sulforaphane isn't just pulling the fire alarm, it is actually rewriting the building codes of the cell.
Yes, that brings us to its role as an HDAC inhibitor.
Okay, unpack that for us.
So to understand this, think of your DNA as the hardware of a computer, and epigenetics as the software that tells the hardware what to do. HDAC stands for histone deacetylase.
It has a mouthful.
It is, but essentially it is an enzyme that cancer cells often hijack. Cancer uses HDACs to silence or turn off the body's natural tumor suppressor genes.
So the mutated cell basically hides the genetic instructions that would normally tell it to self-destruct before it turns into a tumor.
Yes.
So the cancer cell throws a thick, dark blanket over the genetic code that can stop it from spreading.
Perfect analogy. And sulforaphane acts as the biochemical hand that rips the blanket off.
Oh, wow.
By acting as an HDAC inhibitor, sulforaphane blocks the silencing enzyme. It works on the epigenetic level to help re-express or turn those protective tumor suppressor genes back on.
It reboots the antivirus software.
Exactly. This molecular mechanism provides a deep, satisfying explanation for what epidemiologists have observed for decades: that human populations with a consistently high intake of cruciferous vegetables show significantly reduced risks of prostate, breast, lung, and colorectal cancers.
It is incredible to see the epidemiological data line up perfectly with the cellular mechanics, and the metabolic protection is so incredibly broad. Like, there was an animal study in our sources that completely blew my mind.
Oh, the high glycemic one.
Yes. Researchers took mice and fed them a high glycemic index diet.
Yeah.
Essentially, they fed them the laboratory equivalent of a terrible junk food diet.
Right.
The kind of high-sugar, highly processed food designed to rapidly make them obese and diabetic.
And that high glycemic index study is a powerful demonstration of sulforaphane's metabolic leverage. In that study, the mice fed the junk diet, but who were also given sulforaphane, showed a 52% reduction in weight gain compared to the mice on the exact same terrible diet who didn't get the compound.
A 52% reduction in weight gain while eating junk food. That is crazy. And the benefits weren't just on the scale, right?
No, they weren't. It fundamentally shielded their internal metabolism from the toxic effects of the sugar. The sulforaphane group maintained much better insulin sensitivity.
How did it do that?
Well, at the molecular level, the researchers found higher levels of crucial insulin receptor proteins, specifically IRS1 and IRS2.
Okay, think of these proteins as the doorknobs on the outside of a cell. When insulin turns the doorknob, sugar is allowed inside to be burned as energy. The sulforaphane basically kept those doorknobs functioning.
That's a great way to visualize it. They also showed an upregulation of a gene called SRT1.
What does that one do?
URSUT acts as a metabolic traffic cop. When it is activated, it tells your cells to stop storing excess calories as fat and start burning them for energy. The sulforaphane was actively directing their metabolism away from disease.
Okay, I have to jump in here and offer some heavy pushback on behalf of the listener.
Go for it.
This all sounds miraculous. It protects the brain, it manages blood sugar, it stops cancer from hiding.
Yeah.
But what is the practical application for you listening to this right now?
That is the big question.
Right. Do you have to quit your job, buy a farm, and spend all day chewing on pounds of raw, unseasoned broccoli sprouts just to get these benefits? Because nobody is going to actually do that.
No, absolutely nobody is going to do that. It is the ultimate practical question. And thankfully, food chemists have provided a highly accessible hack that is currently being utilized in clinical trials.
A hack? Tell me more.
We know that heat destroys the myrosinase enzyme in your broccoli. Right, leaving the glucoraphanin stranded and inert. But myrosinase isn't exclusively found in broccoli. It is present across the entire Brassica family of plants.
Wait, really? Where else is it hiding?
One of the most concentrated, shelf-stable, and easily accessible sources of active myrosinase is common, everyday mustard seed powder.
You mean the kind you can buy in the spice aisle for $2?
Exactly that kind.
Yeah.
Researchers have tested what happens when you take thoroughly cooked, chemically dead broccoli and simply sprinkle a small amount of ground mustard seed powder over it on your plate.
Oh my gosh. The glow stick hack. You are adding the missing chemical back in.
Precisely the glow stick hack. The dried mustard seed powder provides the fully active myrosinase enzyme.
So it's still alive in the powder.
Yes. When you eat the meal, the powder hits the moisture of the cooked broccoli in your stomach, and it acts as the spark. It interacts with the surviving glucoraphanin that was trapped in the cooked broccoli, and it successfully reactivates the violent chemical reaction.
Produces sulforaphane right there in your digestive tract.
Exactly.
That is brilliant. You can have your deeply roasted, heavily seasoned, delicious broccoli, sprinkle a tiny pinch of mustard powder on it before you eat, and you get the entire superpower back.
It is a wonderful, highly practical application of food chemistry. However, I must add a vital note of scientific caution here.
Fair enough. Let's hear it.
Sulforaphane is a remarkably powerful molecule, but it is not a magic bullet.
Right. You can't just eat donuts every single day, take a broccoli extract pill, and expect to live forever.
Dr. Rosengren was explicitly clear about this in his findings. Lifestyle factors like consistent exercise, overall healthy eating, and weight management, they must remain the absolute foundation of any treatment for metabolic conditions like prediabetes.
There are no shortcuts.
None. Furthermore, we have to respect the pharmacology of these highly active compounds. Activating the NRF2 pathway is beneficial, but you do not want the fire alarm ringing permanently.
Because the cell would spend all its energy defending itself instead of just doing its normal job.
Exactly. In laboratory rat studies, administering massively high doses of sulforaphane, around 150 to 300 milligrams per kilogram of body weight, well, it caused severe toxicity.
Oh, wow. What happened to them?
The animals experienced heavy sedation, dangerous hypothermia, and extreme muscle weakness. Essentially, the massive overstimulation caused their bodies to start shutting down non-essential systems.
That sounds intense.
It is. Now, those doses are wildly higher than anything you could ever physically consume from a normal dietary intake of broccoli or sprouts. But it proves a fundamental principle of toxicology. The dose makes the poison.
Right. More isn't always better.
Precisely. It highlights exactly why rigorous clinical trials are so necessary to determine the safe therapeutic windows for humans, and why the specific source and dose of any isolated supplement truly matters.
So, as we wrap up this deep dive into the CRISPR drawer, let's recap the incredible journey we've been on. First, we discovered that sulforaphane is a remarkably potent cellular protector, but it is locked inside cruciferous vegetables as a harmless precursor.
And it absolutely requires the myrosinase enzyme, the biological snap of the glow stick, to be activated, an enzyme that is incredibly sensitive to the heat of your stove.
Second, we explored the groundbreaking clinical research proving that once activated, sulforaphane does not just act as a simple sponge for toxins. It pulls the genetic fire alarm, activating the body's internal NRF2 defense system.
This targeted epigenetic mechanism is showing immense clinical promise in managing prediabetes, shielding the body from metabolic disease, and even fundamentally repairing the cellular environment of the brain to improve the negative symptoms of complex psychiatric conditions like schizophrenia.
It fundamentally shifts your perspective on what is sitting on your plate. Food is not just caloric fuel. That is the massive big picture insight here.
It truly changes the paradigm. A calorie is not just a unit of energy to be burned. The food we eat is a highly complex, deeply intricate chemical instruction manual. The specific molecules we consume, chew, and digest are actively communicating with our DNA in real time. They are shifting our epigenetic markers, having a dialogue with our gut microbiome, and fundamentally altering our biology on a cellular level every single day.
It is absolutely mind-blowing when you really think about the mechanics of it. And it leaves me with one final provocative thought for you to take away today. If a simple, everyday vegetable, like the humble broccoli sprout, holds the chemical key to unlocking our body's own internal defense systems, rewriting our epigenetic code, and altering the very chemistry of our brains, what other chemical secrets are hiding in plain sight in the produce aisle, just waiting for the exact right enzyme to wake them up? Think about that the next time you're staring down a plate of mushy vegetables. Thank you for joining us here at the Deep Dive Lab. Be sure to follow and subscribe for more fascinating science stories. We will see you next time.